There is a question that sits underneath every other question in biology. A question so deep and so strange that most biologists spend their careers deliberately not thinking about it, because thinking about it too hard makes it hard to think about anything else. The question is not how evolution works, because we know how evolution works. It is not how the genetic code operates, because we know how the genetic code operates. It is not how cells divide, or how proteins fold, or how the immune system distinguishes self from non-self. We know all of these things, in extraordinary detail, built up over a century and a half of biological research that has produced one of the most comprehensive and internally consistent bodies of knowledge in human history. The question underneath all of this, the question that all of it presupposes the answer to, is the question of how the first cell came to exist, how life began, how chemistry became biology, how the dead became alive. This is not a question biology has answered. It is a question biology is still asking, with increasing urgency and increasing ingenuity, and the answers that have been proposed in the last 3 decades are extraordinary enough and plausible enough that the question no longer feels unanswerable. But it has not been answered. And the distance between the answer and where we currently are is significant enough that intellectual honesty requires acknowledging it, even in the middle of what has been remarkable scientific progress. The difficulty of the question comes from a specific and devastating circularity that sits at the heart of every living system we know about. Life, as we find it on Earth, requires 3 things simultaneously: information storage, catalysis, and a boundary. Information storage is performed by DNA, the long double-stranded molecule that encodes the instructions for building every protein in the cell, that is copied every time the cell divides, that carries the evolutionary history of the organism in its sequence of base pairs. Catalysis, the acceleration of chemical reactions to the rates that life requires, is performed by proteins, specifically by enzymes, which fold into precise 3-dimensional shapes that allow them to bind specific molecules and facilitate specific chemical transformations with extraordinary specificity and speed. The boundary is the cell membrane, the lipid bilayer that separates the interior of the cell from the exterior, maintaining the concentration gradients and the chemical environment that life requires. The problem, the circularity, is that each of these 3 things requires the other 2 to exist. DNA is copied by proteins. Proteins are made by reading the information in DNA. DNA cannot make copies of itself without proteins. Proteins cannot be made without DNA. And both DNA and proteins exist inside cell membranes, which are themselves made by proteins encoded in DNA. The 3 components of life are mutually dependent in a way that seems to make the origin of any one of them without the others impossible. Life looks, from the outside, like something that could not have started, like a machine that requires itself to build itself, like a system that could only arise fully assembled, which it could not have been because nothing assembles things fully assembled from nothing. This is the origin of life problem, stated in its sharpest form. And it is why the question was, for most of the history of science, considered either metaphysical or theological rather than scientific. If life cannot arise without life, then the first life is either a miracle or an eternal fact, either created by something outside of nature or simply always having existed. The possibility that life arose from non-living chemistry through a process of increasing complexity and self-organization, the possibility that the circularity had a beginning, that there was a simpler system from which the current complex system evolved, was not taken seriously as a scientific hypothesis until the 20th century, and was not investigated experimentally until the middle of the 20th century, and is still not fully worked out as a complete and testable theory. The experiment that changed everything, or that seemed to change everything, before the complications became apparent, was performed in 1953 by a graduate student named Stanley Miller working in the laboratory of Harold Urey at the University of Chicago. Miller was 22 years old. He had heard Urey give a lecture about the primitive atmosphere of the early Earth, an atmosphere that Urey believed was reducing, rich in hydrogen and methane and ammonia without free oxygen. And he had an idea. If you put the gases of the primitive atmosphere in a flask with water and pass electrical sparks through the mixture, simulating lightning, you might be able to make organic molecules. Not simple organic molecules, complex ones, the building blocks of life. He set up the apparatus, a flask of water to simulate the ocean, a mixture of methane, ammonia, and hydrogen to simulate the atmosphere, electrical sparks to simulate lightning, a condenser to cycle the gases through the water continuously. He ran the experiment for a week, And when he analyzed the products, he found amino acids, not one or two, multiple amino acids, including glycine and alanine and aspartic acid and glutamic acid, amino acids that are among the most common in proteins in every living organism on Earth today. From simple gases and electricity and water, in 1 week he had made the building blocks of proteins. He had made life's ingredients from scratch. The announcement was on the front page of newspapers around the world. The Miller-Urey experiment was widely interpreted as having solved, or nearly solved, the origin of life problem. If the building blocks of life could form so easily from simple chemistry, if amino acids practically made themselves given the right conditions, then the origin of life seemed like it might be a relatively straightforward chemical process, something that followed naturally from the chemistry of the early Earth. The optimism was premature, but the experiment itself was genuinely transformative. It established for the first time experimentally that the boundary between chemistry and biochemistry was not absolute, that the molecules of life could form from simpler molecules by processes that required no special intervention, no divine hand, no mystery beyond ordinary chemistry. This was a conceptual revolution, even if it was not a complete solution. The complications accumulated over the following decades. The composition of the early Earth's atmosphere turned out to be more uncertain than Urey had assumed. Current models suggest it was probably less reducing than methane and ammonia rich, more dominated by carbon dioxide and nitrogen, which are less productive of amino acids under Miller-Urey type conditions. The amino acids made by the Miller-Urey experiment are racemic, equal mixtures of left-handed and right-handed versions, while life uses almost exclusively left-handed amino acids, a selection that somehow had to occur on the early Earth, but whose mechanism is unknown. And even if you have all the amino acids you want, getting them to link together into proteins of specific sequences in water is not straightforward. The polymerization reaction that links amino acids is thermodynamically unfavorable in water because it releases water, and in a water environment the equilibrium strongly favors the broken-down monomers over the polymerized chains. Making proteins from amino acids in water requires you to fight the thermodynamics, which requires energy, which requires, in the living cell, other proteins to catalyze the reaction. The circularity reasserts itself. The deeper problem with starting from amino acids in proteins is that proteins by themselves cannot carry hereditary information. Proteins are extraordinarily capable catalysts, but the information needed to make a specific protein, the sequence of amino acids that folds into the specific shape needed for a specific function, is not stored in the protein itself. It is stored in the sequence of nucleotides in the gene that encodes it. To get heritable, evolvable chemistry, chemistry that can respond to natural selection, that can improve over generations, that can develop increasing complexity through the accumulation of beneficial changes, you need both catalysts and information. Proteins provide catalysts but not information storage. DNA provides information storage but not catalysis, and neither can arise without the other. In the biology we know, this impasse, the chicken and egg problem of DNA and proteins, is what made the RNA World hypothesis so compelling when it was first articulated clearly in the 1980s, and so central to origin of life research in the decades since. The hypothesis is this: before there were DNA and proteins, there was RNA, ribonucleic acid, the molecule that in modern cells serves as the intermediary between DNA and protein, the messenger that carries the genetic information from the nucleus to the ribosomes where proteins are made. RNA is structurally similar to DNA. It is also a long chain of nucleotides, with a sequence that carries information, but it is single-stranded rather than double-stranded, and it uses uracil instead of thymine as one of its 4 bases. And critically, RNA can do something that DNA cannot. It can fold into complex 3-dimensional structures and act as a catalyst. The discovery of catalytic RNA, ribozymes as they are called, was made in the early 1980s independently by Thomas Cech at the University of Colorado and Sidney Altman at Yale University, and it won both of them the Nobel Prize in Chemistry in 1989. Cech found that the RNA molecule in a single-celled organism called tetrahymena could catalyze its own splicing, could cut itself and rejoin itself without any protein enzyme, using only the RNA molecule itself as the catalyst. Altman found that the RNA component of an enzyme called ribonuclease P was the catalytically active part, that RNA, not protein, was doing the work. These discoveries shattered the dogma that enzymes were always proteins. RNA could fold into shapes that allowed it to catalyze chemical reactions. RNA was both an information-carrying molecule and a potential catalyst. RNA could, in principle, do both jobs simultaneously. The RNA world hypothesis puts these two properties together into a scenario for the origin of life that breaks the circularity. In the RNA world, the first self-replicating system was made of RNA. RNA molecules that could catalyze their own copying, ribozymes that acted as RNA polymerases, copying RNA from RNA, could have arisen on the early Earth and could have begun evolving through natural selection. Variation would arise from copying errors, random mutations introduced during replication. Selection would favor RNA molecules that were better at replicating themselves, that copied more accurately, that replicated faster, that were more stable in the early Earth environment. The RNA molecules would evolve, becoming more sophisticated over time, eventually developing the capacity to catalyze the synthesis of peptides, short chains of amino acids, using the information in their own sequence. The peptides would improve catalysis, making the RNA replication more efficient. Eventually, DNA would appear, a more stable information storage molecule, and the division of labor we see in modern cells would emerge. DNA for information storage, RNA for information transfer, proteins for catalysis. The RNA world would be replaced by the DNA-RNA protein world we inhabit. This is a beautiful and compelling story. It is also, in its details, extremely hard to make work, and the people who have spent their careers trying to work out the details are the first to say so. The central challenge is getting nucleotides, the building blocks of RNA, to form on the early Earth, and then getting them to link together into RNA chains of sufficient length to have catalytic activity, and then getting those chains to fold into structures that can actually catalyze replication. The formation of nucleotides is not straightforward. A nucleotide consists of three parts: a sugar, ribose, in RNA, a phosphate group, and a nitrogenous base, adenine, guanine, cytosine, or uracil. Ribose can form from formaldehyde through a reaction called the Formose reaction, but the Formose reaction produces a complex mixture of many different sugars, of which ribose is a minority component, and the selection of ribose specifically from this mixture is not easily achieved by known chemistry. The bases can form from simpler molecules. Adenine was famously synthesized from hydrogen cyanide by Juan Oro in 1960, and other bases have been synthesized under plausible prebiotic conditions, but linking a base to ribose to form a nucleoside, and then adding a phosphate to form a nucleotide, without the enzymes that perform these steps in modern cells, has been extraordinarily difficult chemically. For decades, the synthesis of pyrimidine nucleotides, the nucleotides containing cytosine and uracil, under plausible prebiotic conditions seemed essentially impossible, and the difficulty seemed like a fatal problem for the RNA world hypothesis. And then, in 2009, a chemist named John Sutherland at the Medical Research Council Laboratory of Molecular Biology in Cambridge, England, published a paper that transformed the field. Sutherland and his colleagues had found a new chemical pathway to cytidine, the nucleoside containing cytosine, that bypassed the steps that had seemed impossible. The key insight was that the standard approach, trying to attach a preformed cytosine base to a preformed ribose sugar, was the wrong way to think about the chemistry. Instead, Sutherland's pathway assembled the nucleotide in a completely different way, building up the sugar and the base simultaneously from simpler starting materials, through a series of reactions that proceeded efficiently under conditions plausible for the early Earth. The pathway started from cyanamide, cyanoacetylene, glycolaldehyde, glyceraldehyde, and inorganic phosphate, molecules that could all have been present on the early Earth, and produced cytidine 2-flumer-3,4 cyclic phosphate, a form of the nucleotide that could be converted to the standard form under simple conditions. Sutherland's synthesis was not just a clever chemical trick, it was a conceptual breakthrough. It showed that the RNA nucleotides could form under prebiotic conditions, that the chemistry of the early Earth was capable of producing the building blocks of the RNA world. The synthesis was not the only possible pathway, and it was not yet a complete picture. The synthesis of purine nucleotides and their integration with pyrimidine nucleotides into a coherent prebiotic chemistry remained to be worked out, but it broke the logjam. The impossible had become possible. The building blocks of RNA could form from simple chemistry. The RNA world hypothesis gained a solid chemical foundation that it had previously lacked. Sutherland's work opened a research program that has been enormously productive in the 15 years since. His group and others have worked out chemical pathways from simple precursors, hydrogen cyanide, formaldehyde, water, and a few other simple molecules, to not just RNA nucleotides but to amino acids and lipid precursors simultaneously, from the same starting materials, in the same chemical environment. This is a striking result. The building blocks of the three major classes of biological macromolecules, nucleic acids, proteins, and membranes, can all be derived from the same simple chemistry, suggesting that they might all have appeared together on the early Earth, potentially solving the problem of how the three components of a cell arose simultaneously. The vision of a single, interconnected prebiotic chemistry that produces all the ingredients of life from simple starting materials, hydrogen cyanide, sulfur, water, and light, is one of the most exciting recent developments in origin of life research. But making the building blocks of RNA is not making RNA. The next step, polymerizing nucleotides into RNA chains, has its own challenges. In water, nucleotides do not spontaneously link together into long chains. The phosphodiester bond that links nucleotides in RNA is thermodynamically unfavorable in aqueous solution, for the same reason that amino acids do not spontaneously polymerize into proteins. Energy input is required. In modern cells, the energy comes from ATP, which is itself a product of protein-catalyzed reactions. On the prebiotic Earth, an alternative energy source was needed. Possible sources include wet-dry cycles, the repeated wetting and drying of pools on the early Earth's surface, which could concentrate nucleotides and drive polymerization during the dry phase, and mineral surfaces, which could absorb nucleotides and orient them in ways that promote polymerization. Experiments have shown that RNA chains of 30 to 40 nucleotides can be produced on clay mineral surfaces, and that these chains can even have catalytic activity. The polymerization of RNA under prebiotic conditions is not solved, but it is no longer obviously impossible. The most fundamental challenge remaining for the RNA world hypothesis is the self-replication problem. The entire scenario requires RNA molecules that can copy RNA, ribozymes that act as RNA polymerases, reading a template RNA and synthesizing a complementary copy. This is the function of the enzymes that copy RNA in modern cells, and those enzymes are proteins, not RNA. Does an RNA molecule exist, or can one exist, that can perform this function? This is the central experimental question of the RNA world, and it has been the focus of an enormous amount of research, primarily through the technique of directed evolution, the laboratory equivalent of natural selection applied to molecules. The pioneer of directed evolution applied to RNA is Gerald Joyce, who has spent decades at the Scripps Research Institute in La Jolla, California, developing ribozymes of increasing catalytic capability through repeated cycles of mutation and selection. Joyce's group and others have evolved ribozymes that can copy short RNA sequences, that can catalyze a range of chemical reactions, and that can in some cases undergo exponential amplification when supplied with the right components. In 2009, Joyce's group reported a ribozyme that could replicate itself, that could copy its own sequence, though under highly constrained conditions that are not plausible for the prebiotic Earth. In 2012, his group reported a pair of RNA molecules that could cross-replicate each other, each copying the other, which is a closer analog to what an RNA world organism might have looked like. In 2021, Philip Holliger's group at the MRC Laboratory of Molecular Biology in Cambridge demonstrated a ribozyme that could copy RNA sequences of up to 200 nucleotides, longer than any previous RNA copying ribozyme, using a completely different catalytic mechanism from previous ribozymes. These are genuine advances. The experimental demonstration that RNA can in principle copy RNA, that ribozymes with this function exist, at least under laboratory conditions, validates the central claim of the RNA world hypothesis. But the ribozymes developed in these experiments are themselves products of directed evolution in the laboratory, which means they have been optimized by a process that is designed and controlled by intelligent researchers. The question of whether ribozymes of comparable capability could arise spontaneously on the early Earth, without directed evolution, without intelligent design, through the random chemistry of a prebiotic environment, that question has not been answered. The gap between what can be produced by sophisticated laboratory evolution and what could arise spontaneously by chance remains significant, though the gap has narrowed considerably. The RNA world hypothesis is not the only serious proposal for the origin of life, and the alternatives illuminate aspects of the problem that the RNA world does not address well. One of the most important alternatives, or rather complements, is the metabolism first hypothesis, which argues that the first living systems were not replicating RNA molecules, but were networks of chemical reactions that together constituted a primitive metabolism, a system that extracted energy from the environment and used it to maintain itself and grow. The proponents of this view argue that replication came later, after metabolism had established a chemical system capable of supporting it, and that the RNA world hypothesis puts the cart before the horse by focusing on replication and neglecting the energy problem. The most sophisticated and most scientifically compelling version of the metabolism first hypothesis centers on alkaline hydrothermal vents, a specific type of seafloor vent discovered in the early 2000s that is completely different from the hot, acidic hydrothermal vents near mid-ocean ridges that are often cited in popular accounts of the origin of life. Alkaline hydrothermal vents. The most famous example is the Lost City vent field in the Atlantic Ocean, discovered in 2000 by a team led by Deborah Kelly, produce warm, alkaline water rich in hydrogen gas and other reduced molecules, and they are riddled with tiny, interconnected mineral pores of exactly the size of cells. The scientist who has most forcefully championed these vents as the cradle of life is Mike Russell, a geochemist who has worked for decades at the boundary of geology, chemistry, and biology, and whose ideas about the origin of life are among the most carefully thought out and most chemically grounded in the field. Russell's argument begins with energetics. Every living cell must extract energy from its environment to do the chemical work of maintaining itself and reproducing. In modern cells, much of this energy comes from the movement of protons across membranes. The electrochemical gradient of protons drives the synthesis of ATP, the universal energy currency of life, through a protein complex called ATP synthase. This proton gradient, maintained by active pumping by membrane proteins, is one of the most ancient and most universal features of life. It is found in every domain of life: bacteria, archaea, eukaryotes, which means it was present in the last universal common ancestor, the organism from which all life on Earth descended. The universality of the proton gradient suggests that it is not a later invention, but a fundamental feature of life from its earliest beginnings. Russell points out that alkaline hydrothermal vents naturally produce proton gradients. The vent fluid is alkaline, high pH, low proton concentration. The early ocean, in contrast, was mildly acidic, lower pH, higher proton concentration. The interface between the alkaline vent fluid and the acidic ocean, across the thin mineral walls of the vent pores, constitutes a natural proton gradient, a gradient that could drive chemistry without any pumping, without any membrane proteins, simply by exploiting the electrochemical difference that the geology provides for free. The vent pores, with their iron and nickel sulfide mineral walls, could have provided catalytic surfaces for the chemistry of early metabolism, the reduction of carbon dioxide to organic molecules using the hydrogen provided by the vent fluid. The cell membrane in this picture did not come first. It came later, replacing the mineral walls of the vent pores once chemistry had progressed to the point where a lipid membrane could do the same job. The first life did not need to invent a proton gradient or a membrane. It inherited them from geology. This is a beautiful and compelling framework, and it connects the origin of life to specific existing geological environments that can be studied and characterized. The Lost City vent field in the Atlantic is still active. It has been running for at least 30,000 years and possibly much longer, and it can serve as a model for the conditions under which life might have originated. The chemistry of the vent is not identical to the early Earth, because the early ocean was different from the modern ocean, and the early atmosphere was different from the modern atmosphere, but the physical principles are the same. The vent provides energy gradients, catalytic mineral surfaces, warm temperatures, and the carbon and hydrogen needed for organic synthesis. It is, in Russell's view, a geochemical machine that was pre-adapted to produce life. The most remarkable recent development in the Russell school of thought is the experimental work of Nick Lane, a biochemist at University College London, who has collaborated with Russell and who has spent years trying to reconstruct the chemistry of alkaline vent systems in the laboratory. Lane and his colleagues have built laboratory analogs of alkaline hydrothermal vents, reactors in which alkaline fluid and mildly acidic fluid are brought into contact through iron-nickel sulfide membranes, and have studied the chemistry that occurs at the interface. They have demonstrated that the proton gradient across the membrane can drive the reduction of carbon dioxide to organic molecules, specifically to formate and acetate, simple organic acids that are intermediates in the metabolism of ancient microorganisms called methanogens and acetogens. These are the first steps in a metabolic pathway, the Wood-Lungdahl pathway, that is found in some of the most ancient living organisms, and that is widely considered to be the most ancient metabolic pathway on Earth. A demonstration that vent chemistry can drive the first steps of this ancient metabolism, under conditions plausible for the early Earth, is a significant experimental advance. It does not prove that life originated at alkaline hydrothermal vents, but it demonstrates that the chemistry is right, that the energy gradients, the catalysts, and the substrates are all present, and that they interact in ways that produce the first organic molecules of life's most ancient metabolism. Lane's work, combined with Russell's theoretical framework, has given the alkaline vent hypothesis a chemical and experimental foundation that was previously lacking. The tension between the RNA World hypothesis and the metabolism first hypothesis is one of the central debates in contemporary origin of life research, and it has sometimes been conducted with more heat than the evidence warrants. The two camps have occasionally talked past each other, the RNA World researchers focusing on the problem of replication and largely ignoring the energy problem, the metabolism first researchers focusing on the energy problem and largely ignoring the problem of heredity and evolution. In recent years, there has been a growing recognition that the two approaches are not mutually exclusive, that a complete origin of life scenario requires both energy metabolism and heritable information, and that the question is not which came first in some absolute sense, but how the two came together in a self-reinforcing system. Some researchers are trying to bridge the gap, to find chemistry that is simultaneously metabolic and informational, that produces both the energy-extracting functions of metabolism and the heredity-enabling functions of nucleic acids. One of the most interesting attempts at this synthesis is the work of Sarah Walker, a physicist at Arizona State University who thinks about the origin of life from the perspective of information theory and complex systems. Walker argues that the key to understanding the origin of life is the origin of what she calls instructed chemistry, chemistry that is guided by information, where the information specifies which reactions occur and in what order. In ordinary chemistry, reactions happen according to the laws of thermodynamics and kinetics without any reference to a stored sequence. In life, reactions are instructed by the sequence of nucleotides in genes. The information in the genome tells the cell which proteins to make, and therefore which catalysts are available, and therefore which reactions occur. The transition from uninstructed to instructed chemistry is, in Walker's view, the key transition in the origin of life. Walker's framework connects to a broader perspective on life as a phenomenon that is more about information than about chemistry, a perspective shared by several theorists who have been influenced by the work of Gregory Chaitin, Christoph Adami, and others who have applied information theory to biology. In this view, what makes life different from non-life is not the specific molecules it uses. There is nothing chemically unique about the atoms in a cell, but the information content of its organization, and specifically the fact that the information is self-referential. The genome contains instructions for the molecular machines that read and copy the genome. The origin of life is the origin of this self-reference, the origin of chemistry that describes itself. This is a more abstract formulation of the circularity problem, and it does not immediately suggest solutions, but it may point toward what kind of solution is needed. The question of where life originated, on the surface of the early Earth, in warm ponds or tidal pools or freshwater lakes, or in hydrothermal vents at the bottom of the ocean, is not settled, and different origin of life scenarios favor different environments. Sutherland's chemistry, which produces nucleotides from hydrogen cyanide and other simple molecules, works best in environments that receive ultraviolet light, which means surface environments, not deep-sea vents. The conditions he requires, including sulfur chemistry driven by UV light and wet-dry cycles, are more consistent with a land or coastal environment than with the deep ocean. Russell's alkaline vent chemistry, by contrast, is explicitly a deep-sea scenario, driven by geological energy rather than solar energy. Darwin himself, in a famous letter written in 1871, suggested that life might have originated in a warm little pond, with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc. present, a surface environment, exposed to light and energy from multiple sources. The warm little pond concept has been developed and refined over the years, and experiments with simulated warm pond conditions have produced some encouraging results. Jack Szostak, a Nobel Prize-winning chemist who spent years at Harvard and who has now moved to the University of Chicago, has been one of the most creative and productive researchers in this tradition. Szostak's approach is bottom-up. He is trying to build a simple protocell in the laboratory, from scratch, using the kinds of molecules and conditions plausible for the early Earth. His group has made extraordinary progress on the membrane problem, demonstrating that fatty acids, which are simpler than the phospholipids that make up modern cell membranes, can spontaneously form vesicles, closed spherical shells that mimic the structure of cells, and that these vesicles can grow and divide in the absence of any protein machinery, driven purely by physical forces. They have shown that RNA can be encapsulated in these vesicles. They have shown that the vesicles can take up nucleotides from the surrounding solution. They have explored conditions under which RNA chains inside the vesicles might be able to copy themselves. Szostak's protocell research is enormously important because it attacks the boundary problem, the problem of how the interior of the first cell was separated from the exterior, with experimental rigor. He has shown that this problem is not as difficult as it might seem, that fatty acid membranes are astonishingly capable without any protein assistance, and that the conditions under which they form and grow and divide are compatible with a wide range of prebiotic environments. His work suggests that the membrane component of the first cell was not the rate-limiting step, that membranes of sufficient sophistication can arise spontaneously under reasonable conditions, and that the harder problems are the chemistry of RNA synthesis and replication inside the membrane. There is a specific and important line of research that connects the origin of life to chemistry happening right now, in the universe, in the molecular clouds between the stars and on the surfaces of asteroids and comets. Astrochemistry, the chemistry of the interstellar medium, has revealed that the chemistry of carbon is extraordinarily rich and diverse in space, that the molecular clouds from which star systems like ours form are filled with complex organic molecules, including some of the building blocks of life. Glycine, the simplest amino acid, has been detected in cometary dust samples. Sugars, including ribose, have been detected in meteorites. Nucleobases, the information-carrying components of RNA and DNA, have been found in carbonaceous meteorites. The Stardust mission, which collected samples from Comet Wild 2 in 2004, found glycine in the return samples, confirming that amino acids can form in cometary environments. The Rosetta mission, which orbited Comet 67P/Churyumov-Gerasimenko from 2014 to 2016, found organic molecules of extraordinary diversity in the comet's coma, including glycine, phosphorus, and molecules related to the bases of RNA and DNA. The relevance of these discoveries to the origin of life is twofold. First, they demonstrate that the chemistry of life's building blocks is not unique to the Earth. It is happening throughout the universe, on comets and in molecular clouds and in the gas surrounding young stars. The ingredients of life are produced abundantly by ordinary astrophysical chemistry. Second, they raise the possibility that some of the building blocks delivered to the early Earth by meteorites and comets. The bombardment of the early Earth was intense, and enormous quantities of organic material were delivered, contributed to the prebiotic chemistry that eventually led to life. The early Earth was not starting from scratch with simple inorganic molecules. It was seeded with a rich organic chemistry from space. This connects to a broader point about the origin of life that has become increasingly apparent in recent years. The chemistry that produces life's building blocks is not fragile or exotic. It is robust and widespread. Amino acids form in interstellar clouds and on comets and in meteorites and in laboratory simulations of the early Earth. Nucleotides, or their precursors, form under plausible prebiotic conditions from simple starting materials. Lipid membranes self-assemble spontaneously. RNA can fold into catalytic structures. The building blocks of life are products of ordinary chemistry, and ordinary chemistry produces them readily given a few simple ingredients and a source of energy. This does not mean life is inevitable, or that it is common in the universe. The question of how to get from building blocks to a self-replicating system capable of evolution remains the hard part, and we do not know how hard that part is. But it does mean that the starting conditions for life are not rare or improbable. The universe is preloaded with the ingredients. What we do not know is the recipe. The person who has perhaps thought most carefully about the recipe, about the specific chemical and physical conditions under which the ingredients could have come together to produce a self-replicating system, is Lee Cronin, a Scottish chemist at the University of Glasgow, who has developed a framework he calls assembly theory for thinking about the complexity of molecules and its relationship to life. Cronin's central idea is that life can be detected and defined by the complexity of its molecular assembly, the number of steps required to construct a molecule from simple building blocks. Random chemistry produces molecules of low assembly complexity, small, simple molecules that can be made in a few steps. Living chemistry produces molecules of extraordinarily high assembly complexity, large, complex molecules whose construction requires many steps and that could not have arisen by random chance. The boundary between non-living and living chemistry is, in Cronin's framework, a boundary in molecular complexity. Living systems are the ones that produce molecules too complex to have arisen without selection and heredity. This framework has potentially transformative implications for astrobiology, the search for life in the universe. If life can be defined by the assembly complexity of its molecular products, then detecting life on other worlds becomes a question of measuring molecular complexity, which is something that mass spectrometry can do in principle, and which instruments on spacecraft could potentially be designed to measure. Cronin has been developing exactly this idea, in collaboration with Sarah Walker and others, as a potential biosignature for life detection on other worlds. A way of detecting life that does not depend on knowing what specific molecules to look for, but only on measuring the complexity of the molecular mixture. If a planetary environment produces molecules of very high assembly complexity, it is almost certainly doing so because there is a living system producing them. Random chemistry cannot, except with negligible probability, produce the kinds of complex molecules that life routinely makes. Assembly theory also has implications for the origin of life problem. If life is characterized by high assembly complexity, then the origin of life is the origin of the first chemistry capable of producing high assembly complexity, the first chemistry in which selection acts on molecular variations and amplifies the complex at the expense of the simple. The transition from random chemistry to life is the transition from chemistry that is going nowhere in particular, producing the most stable and most abundant simple molecules, to chemistry that is being directed by selection, producing increasingly complex molecules that are better and better at perpetuating themselves. The origin of life is the origin of selection, and the origin of selection requires, at minimum, heredity, the ability of a molecular system to copy itself with variation. Which brings us back to the RNA world, the convergence of different research traditions on this problem, the RNA world chemists, the metabolism first theorists, the astrochemists, the assembly theorists, the protocell researchers, is one of the most encouraging signs that the origin of life problem is genuinely making progress. Different communities are approaching the same problem from different directions, and the approaches are beginning to converge on a picture that, while still incomplete, is more coherent and more chemically grounded than anything available 20 years ago. What the picture looks like, in its current form, is roughly this. The early Earth, about 4 billion years ago, was a world of intense geological activity, with hydrothermal systems both at the surface, in the form of hot springs and geysers, and in the deep ocean, in the form of alkaline and acidic hydrothermal vents. The atmosphere was rich in hydrogen cyanide, produced by the ultraviolet irradiation of nitrogen and carbon-containing gases, and the surface was bombarded by asteroids and comets that delivered additional organic material. Cycling of wet and dry conditions, driven by tidal forces and geological activity, concentrated organic molecules in surface pools and on mineral surfaces. Simple chemical reactions, driven by UV light and mineral catalysts, built up the building blocks of nucleotides, amino acids and fatty acids from simpler precursors. Nucleotides linked together into short RNA chains on mineral surfaces. Some of those chains by chance had sequences that allowed them to fold into structures that could catalyze chemical reactions, including, eventually, reactions that helped copy RNA sequences. The first self-replicating RNA system, encapsulated in a fatty acid membrane that gave it a boundary and concentrated its chemistry, was the first protocell, the last ancestor of all life on Earth. This is a plausible story. It is grounded in chemistry that has been demonstrated experimentally, at least in part. It is not a complete story. There are still steps that have not been worked out in chemical detail, and there are aspects of the story that are contested. But it is no longer a story that requires magic. It is a story that requires chemistry, difficult chemistry, improbable chemistry in the specific sense that it had to happen in the right place at the right time with the right ingredients, but chemistry that follows from the laws of physics and chemistry without any additional assumptions. The question that origin of life research has not yet answered, the question that matters most for understanding whether life is common or rare in the universe, is how improbable the critical steps were. Was the origin of life a near certainty given the right conditions, something that would happen quickly wherever the conditions were met? Or was it an extraordinarily improbable event, something that required enormous amounts of time and enormous numbers of failed attempts before succeeding? We do not know. The fossil record of life on Earth suggests that life appeared very early, within a few 100 million years of the Earth becoming habitable, possibly within a few tens of millions of years. This rapidity has been interpreted by some researchers as evidence that the origin of life is easy, that given the right conditions it happens quickly. Others point out that we should not overinterpret a single data point. We know that life appeared quickly here, but we do not know how many times it tried and failed before succeeding, and we do not know how many other worlds have tried and failed without ever succeeding. The answer to the question of how improbable life is, how difficult the origin of life is, how common life is likely to be in the universe, is one of the most important answers that science has ever tried to find. If life is common, if it arises quickly wherever the conditions are right, then the universe is likely full of it, and the Fermi paradox becomes more urgent, and the discovery of life beyond Earth becomes likely within the coming decades of astronomical observation. If life is rare, if the origin of life is an extraordinarily improbable event, a series of unlikely steps each of which had a small probability, then we may be nearly alone in the observable universe, and the great silence of the sky that Fermi pointed to in 1950 is explained by the rarity of life, rather than by filters that destroy it after it arises. The experiments being done right now, in laboratories in Cambridge and Glasgow and Chicago and La Jolla and Tucson and dozens of other places, are working toward the answer. Not by solving the problem in one dramatic discovery, but by working out the chemistry step by step, constraining the plausible conditions more tightly, determining which pathways are feasible and which are not, measuring the rates of the critical reactions under early Earth conditions, building increasingly sophisticated protocells that approach the complexity of the first true living cells. The progress is real, the problem is hard, the answer is coming, though perhaps not as quickly as we would wish. There is a version of the origin of life story that is deeply unsettling, and it is the version that may be correct. It is the version in which life on Earth arose not from an extraordinarily improbable sequence of events, but from chemistry that was in some deep sense inevitable. Chemistry that the laws of physics and the conditions of the early Earth made likely, perhaps not in the specific molecular details, but in the general outcome. The version in which life is not a miracle and not an accident but a consequence. In which the universe, given the right conditions, generates life the way water generates ice given the right temperature, not with certainty in every instance but reliably, statistically, across a large enough sample of worlds. In which the 400 billion stars of the Milky Way, each with its planetary systems, each providing billions of years of chemical evolution on at least some of its planets, have produced life not once but many times, in many places, in forms we cannot yet imagine. If this version is correct, if life is an inevitable consequence of chemistry given the right conditions, and if the right conditions are not extraordinarily rare, then the universe is full of life. Not just microbial life, but potentially over geological time, complex life, and eventually intelligent life, in numbers that make the Fermi paradox all the more urgent and all the more mysterious. If life is easy, then the silence of the sky is all the more deafening. We do not know yet which version is correct. We do not know if life is inevitable or improbable. We do not know if we are alone or surrounded. We do not know if the chemistry that produced us was a near certainty or a miracle. We are trying to find out. The experiments are running, the theories are being developed, the instruments are being pointed at Mars and at Enceladus and at the atmospheres of exoplanets, looking for the signs of chemistry that has crossed the line from dead to alive. And underneath all of it, underneath the experiments and the theories and the observations, is the question that is personal in a way that no other scientific question is personal. The question of where life came from is the question of where we came from. Not in the evolutionary sense. We know where we came from in the evolutionary sense, and the answer is a long chain of organisms stretching back to the last universal common ancestor, a microorganism that lived about 3.8 billion years ago, in conditions very different from anything alive today. The question is the deeper question. What was before that ancestor? What gave rise to it? What was the moment, if there was a moment, when chemistry became life? When the first molecule began to copy itself and the universe, for the first time in any place we have evidence of, had something in it that was more than just matter following the laws of physics? Stanley Miller did not answer that question when he ran his experiment in 1953, but he did something almost as important. He showed that the question was scientific, that it had a chemical answer, that the gap between chemistry and life, however large it seemed, could be approached from the chemistry side, could be explored experimentally, could be narrowed through patient work, could eventually be bridged. He was 22 years old. He was a graduate student. He was running an experiment that most of his contemporaries thought was a waste of time. He was right. The people doing the work now are also right. Right to pursue the question, right to think it is answerable, right to believe that the chemistry of the early Earth was capable of producing the first living system and that understanding how it did so is within the reach of science. They may be right about more than that. They may be right that the universe is full of chemistry similar to what happened here, that the early Earth was not unique but representative, that the 4 billion years of life that followed that first event of self-replication are not a cosmic accident but a cosmic regularity, happening and rehappening across the galaxy in forms we have not yet found but that are out there, alive, made of the same atoms and the same chemistry as we are, products of the same deep tendency of carbon and water and energy to organize themselves into something that persists and grows and evolves. Or they may be wrong about that. The universe may be silent not because something kills civilizations, but because life is so improbable that we are the only example in the entire observable universe of chemistry that crossed the line. We do not know. We are trying to find out. We have been trying since Stanley Miller sat down at his apparatus in 1953, since Darwin wrote his letter about the warm little pond in 1871, since the question first crystallized into a form that science could approach. The answer is closer now than it has ever been. It is not yet in hand, but somewhere in the chemistry of the early Earth, somewhere in the warm little pond, or the alkaline vent, or the drying beach, or the mineral surface, it started. Something clicked. Something that had never happened before in any place we have evidence of happened, and what had been dead and dumb and purposeless became something that persisted, something that grew, something that made copies of itself, something that, over 4 billion years of evolution, through mass extinctions and continental drift and asteroid impacts and ice ages, eventually became something that could wonder how it had happened. You are that something. You are 4 billion years of chemistry that has learned to ask where it came from. The answer is not complete, but the asking is one of the most extraordinary things that has ever happened in this corner of the universe, and it is happening now, and the people who have dedicated their lives to answering it are getting closer every year. The chemistry is still running. The question is still open. The answer is coming.